Good morning. In today's class, we will see about the electroencephalogram, which is EEG signal. Introduction. The electroencephalogram is a recording of the electrical activity of the brain from the scalp. The first recordings were made by Hans Berger in 1929. The brain waves are the summation of neural depolarization in the brain due to the stimuli from the five senses. So before knowing about the EEG signals, you must know about the characteristics of the EEG signal. So when the EEG signal is measured directly on the brain, the amplitude of the EEG signal will be from about 10 millivolts. When you measure on the scalp, the signal will propagate through the skull, gets attenuated, and the voltage level of the EEG signal will reduce to 1 to 100 microvolts. So if you measure the EEG signal directly on the brain, you will get a higher amplitude signal. When you measure it at the scalp of the brain, scalp of the head, then the potential of the EEG signal reduces to 100 microvolts. The frequency range of the EEG signal varies from 0.5 to 300 Hz. So usually we use from 0.5 to 30 Hz, and the brain waves are classified as alpha, beta, theta, delta and gamma. So how do we measure the EEG signal? We place the electrodes on the frontal lobe, parietal lobe, occipital lobe and the temporal lobe of the cerebral cortex. So now we must know what is a neuron. So nervous tissue is composed of two types of cells, which are the neurons and the glial cells. The neurons transmits nerve messages. The glial cells are in direct contact with the neurons and often surround them. So you can see the picture of the neurons. Our brain has hundreds of billions of them. So this is the neuron network shown here, a group of real neurons. Now what is a neuron? A neuron consists of three parts. One is the input or dendrite, also we call it as receivers, then a cell body and then an axon. So the end of the axon is called as the axon terminal. So you can see the tree-like structures here, which are called as the dendrites. They receive information from the other nearby neurons or axons or the cell body, and then they transmit. They receive the information is received through the dendrite and transmitted to the axon, to the other neurons, nerve cells. So here this is the cell body, otherwise we call it as soma. And this axon, this red color tail of this neuron is called as the axon. This is covered by the myelin sheath. This myelin sheath is an insulating fatty layer that speeds up the transmission of information from the input to the output. And in between the myelin sheath you can see some gap. They are called as node of Ranvier. The node of Ranvier is used to speed up the transmission. So how it will speed up the transmission is, information from the dendrite will come here through this axon. This part is called as the axon hillock. From the axon hillock it gets transmitted to this node, this node, this node. So the information will be jumping from here to here until till the tail of the neuron, which is nothing but your axon terminal. So this condition, jumping condition, is called as the saltatory condition. And this you can see something black dot here, which is called as the Schwann cells. The Schwann cells, they make the myelin sheath. From this Schwann cells you get a structure which will surround the axon, making this myelin sheath. So here you have dendrites, which are nothing but your receivers, cell body or soma. The soma has a nucleus, then axon hillock, where the impulses is originated in the neuron. Then from here you have the axon. Axon is surrounded by the myelin sheath, and you have in between you have nodes of Ranvier, and on the myelin sheath you have the Schwann cells, then axon terminal or transmitters. So this it receive information, transmit through the axon to the other neuron. Next we must know what is the synapse. Junction between two neurons is called as the synapse. The synapse can be anatomically classified or functionally classified. Anatomically you can call it as axosomatic, axon and cell body synapse, axodendritic, axon and dendrite synapse, axonic synapse. Just usually axodendritic synapse will happen. In functional classification, we can say that from the axon hillock, electrical signals will be transmitted and received by the other neuron, or chemicals will be transmitted to transfer the information from the dendrite to the through the axon to the other neuron. Now we must know what is a synapse. Synapse is a junction between two neurons is called as a synapse. So in this picture, this white portion is called as the synaptic cleft. In the synaptic cleft only, the information will get transmitted from the presynaptic neuron to the postsynaptic neuron. So whenever there is a synapse, the portion before that synapse is called as presynaptic neuron. After the synapse, it is called as the postsynaptic neuron. So how the information, this is chemical information transmission. So here you have some neurotransmitters. They are called as excitatory transmitters and inhibitory transmitters. So when excitatory transmitters are your acetylcholine or adrenaline, and inhibitory transmitters are dopamine and serotonin or GABA. So these transmitters are sent in synchronous to balance the condition of the brain. If one is not working and one is working, then there is an abnormality in the brain. So when the excitatory synapse happens, the excitatory transmitter will get transmitted. When the inhibitory synapse happens, inhibitory transmitters gets transmitted. So here the chemicals will come here, information will come here, and here the cleft here it will open. This chemical will be transmitted through the cleft. That will be received by the receiving postsynaptic neuron, and the information will send to the wherever portion it is going to go. So this is how a synapse will happen in the between the neurons. So while measuring the EEG signal, you must just know what is the anatomy of the brain. I'll just tell what are the different lobes in the brain, so that we will be placing the electrodes in different lobes. So you have the frontal lobe, parietal lobe, temporal lobe and occipital lobe. So we will place each part of the lobe functions differently, and each of them are connected to our body. So we place different electrodes here, and then we measure the EEG signal from the brain. So the electrodes and brain signals. So electrode cap contains large number of electrodes, and we use number of electrodes to measure the EEG signal in powers of 2. 2 power 3 is 8, 2 power 4 16, and so on. So we use electrodes in powers of 2, so that more information can be gathered from the brain. So in research studies, 256 electrodes or 512 electrodes will be used to identify or to diagnose particularly in which area the brain is not functioning properly. These are the EEG electrodes. EEG electrodes, you have the input, this portion, it consists of two ends. This is the scalp electrode, which is placed on the brain through a gel. So the place of the brain is cleaned properly, and the gel is fixed along with the scalp electrode. The other end of the electrode is fixed to the preamplifier. So this is the electrode placement. So here we can see that the electrodes can be placed in this particular portion. So how we have to place the electrodes? You have some measurement to be done. So here you measure the distance between the nace ion and the in ion. So nace ion and the in ion, you measure the distance, and A1 and A2 you measure the distance. A1 and A2 represents the auricular electrode, which are the reference electrode that is placed on the ear, on the ear lobes. So auricular electrodes are A1 and A2. Nace ion is the place between the eyebrows and the nose. The intersection of the eyebrows and the nose is your nace ion. In ion is the portion where that small bump is there in the head. Below that bump, you can that portion is called as the in ion. So you measure this distance near ion in ion A1 and A2. Then you will intersect both. That intersection you fix an electrode called CZ. So from in this picture you can see that towards the right of the brain, all the electrodes are numbered even, and to the left of the brain all the electrodes are numbered odd. So always right hemisphere will have the even numbered electrodes and left hemisphere will be numbered in odd. So for example F8, this side F7, F4, F3, like that. And this FP represents prefrontal, F represents frontal, C represents central, P represents parietal, O represents occipital, and T represents temporal. And this Z represents the central electrode. Okay. So now you have measured this distance between S and in ion A1 and A2, fixed up CZ. Now you know this distance from A1 to A2. Take 20% of that distance, fix up this point C4. Then from C4 take 20%, fix up T4. Similarly towards the left hand side, take 20% of the measurement, fix up C3. Then 20% fix up T3. So now you know this T3, C3, C4, T4 are fixed. Similarly from the near ion to in ion, we fix the electrode. From CZ, measure 20%, fix up FZ in the frontal portion, FPZ in the prefrontal portion. From FPZ to near ion, it will be only 10%. Similarly, towards the behind of the head, 20% PZ and 10% OZ. So now you have this electrodes in this manner. In the plus manner, you have fixed up the electrodes. Now we have to fix up the electrodes which are lying in the circular portion. So what we will do, we will connect this OZ through T3 to this FPZ. So connect OZ through T3 to FPZ. Similarly OZ through T4 to FPZ. So when you connect this, you have this from OZ, measure 10%, fix up O1. From O1 20% T5, and from T3 to T5 you will have 20%. Or from T3 take 20%, fix T5. From T5 20% O1. Similarly T4 20% T6, from T6 20% O2. So O1 and O2 the distance will be 20%, and from O1 to OZ it will be 10%, O2 to OZ it will be 10%. Similarly on the other side, T3 20% FZ, F7, from F7 20% FP1. Similarly T4 20% F8, from F8 20% FP2. FP1 and FP2 you will have 20%, or FP1 to FPZ 10% and FPZ to FP2 10%. So now this placement of electrode is over. This one is plus is over and the circle is over. Now we have to fix up the in between electrodes F3, F4, P3 and P4. So what we do is from O2 we would draw a line through C4 to FP2. Similarly FP1 to through C3 to O1. So now this portion you will have. Similarly T5 through PZ to T6, F7 through FZ to F8. So there will be intersection between this line and this line. So there you mark F3, F4, P3 and P4. So now you have fixed up the 16 electrodes onto the scalp plus 2 electrodes A1, A2 and 1 central electrode. So you have 19 electrodes here to measure the EEG signal from the brain. Why it is called as 10-20 international system of electrode? We take 20% and 10% to fix up the distance from the near ion in ion and both the ears to fix up the electrodes. So that is why it is called as 10-20 international system. All throughout the world this measurement is being used for all the persons who are being subjected to EEG signal. So if you want more number of details from the brain, you can add more electrodes and then you can take measurement. So how to take a measurement? A typical EEG session, the patient or subject will be asked to take head bath with shampoo bath, so that there is no dust collected on the scalp of the head. So some subjects, if we need to take data, we can pay them and ask them to sit as for taking data, or students or somebody may come as volunteer. So before taking the data, we have to give them the informed consent form. So we have to inform them what will be the procedure, how will be it will be done, everything. So that informed consent form should be signed from the volunteer or subject before proceeding for EEG recording. Then prepare for electrode placement. So as we have seen in the previous slides, we have to prepare. Sometimes we can use the electrode cap, place the gel over that, and then place the electrodes, or you can measure and then fix up the electrodes. So now the connection between each electrode and the brain is checked. That now the subject is ready for recording. The typical EEG recording session lasts about 1 hour. Generally in hospitals they take 15 minutes of EEG recording. And must know one thing, the person must lie down on the bed in the before taking the EEG recording. In the sitting condition, if you take, you will not get proper EEG recordings. So then now this we have taken the EEG signal. This can be classified as delta, theta, alpha and beta. So delta varies from 0.5 to 4 Hertz, theta from 4 to 8 Hertz, alpha wave from 8 to 13 Hertz, and beta wave greater than 13 Hertz. 14 to 30 Hertz is your beta. So how the waveforms look for different frequency. When it is 13 to 30, it occurs frontally and parietally. We call it as beta. When the person is in the highly excited state, you will get the beta waves. Alpha waves, when a normal conscious condition, the person will be subjected to alpha waves, which is measured occipitally. Occipitally, this is eyes open, eyes closed. So occipital is connected to the eyes. So when the eyes are open, you have a amplitude of about 50 microvolts. When the eyes are closed, the amplitude reduces. When this is sleep, the still more amplitude still more reduces, but the frequency ranges from 8 to 13 Hertz. When the person is sleeping, for a sleeping adult or children up to 8 years, the theta condition is theta wave is 4 to 8 Hertz. For children, this is normal. For sleeping adult, this is normal. And similarly, for sleeping adult, when they go into deep sleep, the signal will go from 0.5 to 4 Hertz. For infants also, the signal will be from 0.5 to 4 Hertz. As they grow up, the information from the signal brain will be come to alpha. For example, suppose if a person is normal, awake, and is having this signal, that means then his brain is having some problem. And this is the condition of epilepsy. You have 3 spikes per second, so it is called as the petit mal. These are the waveforms for sleep stages. During awake, that is 8 to 13 Hertz. Light sleep, this is rapid eye movement. During dreaming, you will get this kind of signal. A deep sleep, a delta. And cerebral death, brain death, you have a straight line. The skull cap and the EEG recorder. This is a recording protocol. You have the electrode placed here. It will be connected to a preamplifier, then a recording system. You will get the EEG like this. So this is taken from Sri Ramachandra Medical University. So this is the EEG transducer recording setup. So this here you will place the electrodes. It will be connected to the channel selector. You can select the channel where you want to measure. Then you have the bus, filter bus, then high pass filter, low pass filter, whereas we have to filter the frequency from 0.5 to 30 Hertz. So for that we use the low pass and high pass filter or band pass filter, and the signal is recorded here. The signal can also be processed and it can be stored in the computer. So here you can see 3 stimulus: visual stimulus, audio stimulus and tactile stimulus. So when the person, we are taking the EEG, they will be subjected to visual stimulus, after some time audio stimulus or tactile stimulus, so that the EEG signal remains normal. For abnormal, when you stimulate and check the EEG signal, the abnormality will be seen apparently when you give a signal. For a normal person, even when a stimulus is given, the signal will be normal. For an abnormal person, if there is a stimulus, the signal can change. So the recorded EEG is shown here. This is FP1A1, FP2A2. So this is monopolar recording. So when with reference to the ear electrodes, if you measure the EEG, it is called as the monopolar recording. This is for during the seizure time, this EEG recording. Here you can see the spikes occurring suddenly it is occurring. And this is a bipolar recording, FP1 F7, F7 T7, T7 P7, like that. So why they take bipolar recording is, so when there is an abnormality in between these two electrodes, we can identify which place where the abnormality occurs. So here the common electrode is F7. So that means that if F7 is not placed properly onto the scalp, or there is a problem near F7, we can easily identify that particular portion. So that is why people go for bipolar recording. Ambulatory EEG system, this is continuous EEG. The patient will be subjected to continuous EEG so that EEG will be studied. Applications of EEG. It is used to find epilepsy. It is used to find the depth of intensity of anesthesia or the anesthetic level. It is used to locate a tumor or during brain injury. After brain injury, the person may be subjected to a tumor. So we can identify the brain injury easily. It is also used to monitor the brain during the brain surgery, and also you can find out the level of consciousness of a person. So if you have any doubts, you can please contact me. Thank you.